Hello friends, warm welcome to all to the channel World of Zoology. This is our lecture series on instrumentation and today's instrument is centrifuge. We'll consider same in our today's lecture with respect to the 3 points. One that is introduction, second one principle and third one is the application. A centrifuge is a device for separating particles from a solution according to their size, shape, density, viscosity of the medium, and the rotor speed. In a solution, particles whose density is higher than that of the solvent, sediment, or sink, and particles that are lighter, it floats on the top. The greater the difference in density, the faster they move. If there is no difference in density, that is isopycnic conditions, the particles stay steady in that particular case. To take advantage of even tiny difference in the density to separate various particles in a solution, gravity can be replaced with much more powerful centrifugal force provided by a centrifuge. This technique plays crucial role in biochemistry or biotechnology as it is non-dispensable part of one or other step in every method involved in biological study, right from separation of the cell organelles to complex experiments involving separation of even subcellular fraction. So we'll consider the two types of centrifuge in our today's lecture, clinical and ultra-centrifuges. Introduction, as it is important to know what is present in biological sample, that is qualitative studies of the biological sample, and how much is present, that is the quantitative analytical techniques such as pH metry, colorimetry, etc. That means here the qualitative as well as quantitative estimation is important. It is often necessary to separate individual components from the given biological sample. General steps in such separation may be considered to follow one or more of the following techniques. It may involve filtration or centrifugation, ultra-centrifugation, flash evaporation, freeze drying, and chromatographic methods. Swedish inventor Gustav de Laval is often credited for the first commercially viable centrifuge machine in 1894. The machine was used to separate cream from the dairy milk. Therefore, we'll consider the principle of the centrifugation, where centrifugal force is used. Centrifugal force can be used to separate lighter particles from heavier portions in a liquid medium. The centrifugal force being greater than the gravitational force can separate the portion of the different or differing densities easily. The centrifugal force acting on the particle is given by the equation centrifugal force F is equal to m omega square r. In this equation, F is the centrifugal force, m is the mass, omega is the angular velocity of the particle, and r is the radius from the center of the circle. The two parameters which are important in centrifugation, one, the revolutions per minute, that is RPM of rotor, and second one, the relative centrifugal force, that is RCF, that is acting on the particles suspended in the solution since the force acting on the sample depends not only on the speed of the rotor but also on its radius. Means the force acting on the sample depends on its radius. Thus, if the sample is spun in two different rotors with the same speed, the different radii, therefore the results would be different. However, if equal RCF, that is relative centrifugal force values, are applied, the sedimentation forces will be identical. As such, the RPM values will be informative only when rotor type or radius is specified. Here, the centrifuge machine is shown in one diagram, and in the another diagram, the part of the centrifuge are shown with the latch, chamber, and motor, that is the rotor, with sample holders, that is test tube holders are shown with motor, and the lid of the, that these are the components of the centrifuge machine. The centrifuge is an essential instrument in cell and molecular biology research. It is generally used to separate biological components based upon their differential sinking, that is sedimentation properties. The four types of centrifuge, that is types of centrifuge, there are four major types of centrifuges. One, low speed or clinical centrifuges, then large capacity refrigerated centrifuge, high speed refrigerated centrifuge, and ultra-centrifuges, out of which only two, that is low speed or clinical centrifuge and ultra-centrifuge, are included in your syllabus. Low speed or clinical centrifuge. The centrifuge machines commonly found in biology or chemistry laboratories are low-speed rotor machines of the about 10,000 RPM, that is revolution per minute, and are used to separate serum plasma from RBCs, that is red blood cells, and to harvest and purify chemical precipitates, intact cells, nuclei, large mitochondria, and large plasma membrane fragments. These are generally used in the laboratories. Low speed or clinical centrifuges. Here the test tube holders which lies on opposite to each other. The sample when placed in the centrifuge machine, they are of the equal volume. Now we'll consider the next, that is ultra-centrifuges. Ultra-centrifuges are machines that are achieve a rotor speed of over 30,000 RPM, even more than that. Minimum speed is 30,000 and above. Refrigeration and high quality vacuum systems are must for these machines. They are used in isolation and purification of membrane components such as endoplasmic reticulum, Golgi membrane, endosomes, ribosomes, DNA, RNA, etc. Here, the fourth one, that is ultracentrifuge, which is included in your syllabus. They are again of the two types. One is the preparative ultracentrifuge, and another is analytical ultracentrifuge. All these centrifuges, which are of the four types, and only two are included in your syllabus. So we have finished the centrifuges with respect to the introduction, principle, and last one, application of the centrifuge. Thank you for watching this video. If you like the video, do like, share the video for the benefit of the maximum students, and subscribe the channel World of Zoology for the notification of my next video on the instrumentation, that is series. and the next topic will be chromatography from instrumentation. Thank you once again.